Hello, Secondary 1 students, with you, Mrs. Sarah Anwar, your biology teacher. Today we will talk about the nucleic acids. Do you know anything about the genes? Yes, it's responsible for transferring the genetic traits from the parents to the offsprings. But how? And what is the structure of the genes? And where we can find it? Okay, let's revise something that we had learned before in Prep 3. It's talking about the cell structure. Okay? This. Okay, if we consider this as an animal cell, so in the center of the cell we can find nucleus, and inside the nucleus something like network called chromatin reticulum. So this is called chromatin reticulum. Okay, if we uncoil the chromatin reticulum, we can find that consists of chromosomes, which take the X shape, and each chromosome consists of DNA coiled around a protein called histone. So this is the protein which is called histone, and this coiled strands are called DNA molecule. So let's summarize it quickly. The cell, inside the cell we have a nucleus, inside the nucleus we have a chromatin reticulum, and the chromatin reticulum is about a number of chromosomes. Each chromosome of them consists of two strands of DNA coiled around each other and coiled around a protein called a histone. So this is the structure till reaching DNA. If we take this DNA strand and unwind it, we can study its structure. As I will explain, DNA is a double helix or two strands coiled around each other like that. And we said that DNA in a polymer consists of monomers. So what is the monomer of the DNA? So it consists of monomers beside each other, monomers to form this polymer. So each one of them, or each monomer in DNA, is called the nucleotide. Nucleotide. So the structure of nucleotide: three parts: sugar and phosphate group, and nitrogenous base. We can draw it like this: a sugar. Two sugar consists of 5 carbon atoms, a phosphate group, and nitrogenous base. If we give the carbon atoms numbers, so nitrogenous base is binding to carbon number 1, carbon 2, carbon 3, carbon 4, carbon 5. So the nitrogenous base binding to carbon number 1 of the sugar, and carbon 5 binding to the phosphate group in the sugar. Here also inside the nucleotide we can find two bonds. The bond that binds the sugar to the nitrogenous base, and other bond which binds the sugar to the phosphate group. Both of them are covalent bonds. So here is a covalent bond, and on the other side covalent bond also. Okay, at your booklet, look at with me. So we talked about that the cell contains a nucleus, and inside the nucleus there is a chromosomes, which structure is DNA, and nucleotides in the building unit. We have two types of nucleic acid: ribonucleic acid RNA, that we didn't talk about it till now, and deoxyribonucleic acid DNA that we are explaining now. Here is the structure of each nucleotide: sugar called the deoxyribose sugar or pentose sugar, which contain 5 carbon atoms. Carbon number 1 binding to the nitrogenous base, carbon number 5 binding to the phosphate group. Here is a covalent bond binding the sugar with the base and binding the sugar with the phosphate group. Okay. Here is the structure of the double helix DNA, or the two strands of DNA. It's like the ladder. If we consider it like the ladder, we have two sides of the ladder and the steps of the ladder. The steps of the ladder are the nitrogenous bases that bind the two strands with each other, while the two sides are the phosphate group and the sugar deoxyribose. Okay, I can draw it to be more clear. Okay, here is the molecule of the DNA double strand. If we unwind the double strand, okay, like that, if we have two strands like this, we can wind them around each other, so it's called as double helix. And if we make unwinding to them, so each strand of them will be separated from each other. So this is one strand, and this is the other strand. I will draw it here. Okay, here is the sugar and the nitrogenous base and the phosphate group. Here is the sugar, nitrogenous base, and phosphate group. Here is the sugar, nitrogenous base, and phosphate group. And also sugar, nitrogenous base, and phosphate group. On the other side, the other strand of DNA, here is the nitrogenous base and the phosphate group. Here is the nitrogenous base and phosphate group. Here is the nitrogenous base and the phosphate group, and so on. So there is one of the strands and the other strand on the other side. The two strands are meeting in the middle by the nitrogenous bases. The nitrogenous bases types are adenine, guanine, thymine, cytosine, and uracil. So we have 5 types of nitrogenous bases that found here between the two strands and binding the two strands with each other. But it's a rule. If you have here adenine, so it must bind to thymine. If you have here thymine, it must bind to adenine. If you have here guanine, it must bind to cytosine, and so on. If we have here cytosine, so it must bind to guanine. And there is a bond that binding the two strands with each other, or binding the nitrogenous bases with each other, which is a hydrogen bond. So don't be confused. We have two bonds till now. We have a covalent bonds that bind all the components of nucleotide together. Here is one nucleotide, the sugar and phosphate and nitrogenous bases. Here is a covalent bond and other covalent bond. And there is a hydrogen bond that binds between the two strands, binding between the nitrogenous bases. So adenine binds to thymine by double hydrogen bond. Thymine binds to adenine by double hydrogen bond. Guanine binds to cytosine by triple hydrogen bond, and so on. So adenine and thymine binds together by double bond, and guanine and cytosine binds together by triple bond. Here we have adenine and thymine. Okay, here inside your booklet. Okay, if you look here, that cytosine consists of one ring, or structure is one ring, and thymine also is one ring. So, does cytosine like thymine? Yes, they are like each other, in shape only, but not in the structure. If they are one structure, there will not be difference between them. But cytosine and thymine consist of one ring. They are similar in shape only, not in the structure. Also, guanine and adenine composed of two rings, but they are similar in the shape only, not in the structure. So adenine must bind to thymine. Guanine must bind to thymine. Okay, here is the two strands of DNA equal around each other. Consider that the two sides, which are in the blue color, are the phosphate group and the sugar, and the nitrogenous bases here in the middle that are binding the two strands with each other and meeting each other by the hydrogen bond. Okay, I think it's more clear. Okay, so return or recall what we have started before. I was asking you about the genes. What are the genes? Yes, the genes responsible for transferring the traits. Or where are the traits? Where are the traits here? The traits represented by the nucleotides or the nitrogenous bases. If we consider, for example, that adenine, thymine, guanine are three nitrogenous bases, this sequence may code for a protein, and this protein will make a character or a trait. So this sequence, by this way, will code it for a protein. This will appear the character or the trait from the parents to offspring. So each three nucleotides with a certain sequence will code a protein for a certain protein. For example, if you have a colored eyes, This colored eye, meaning that you have a certain sequence of nucleotides beside each other, 3 nucleotides beside each other. If you make confusion for this correction, it will give you another protein or another trait or another character. So each 3 of them with a certain sequence will code for a trait. So the DNA must translate it into a protein. Then the protein which makes all of your body will form the trait. Okay, so where is the RNA? Where is the RNA here? Till now, we didn't talk about the RNA. Let's know how we can make RNA from DNA. Here is the cell, and inside the cell, there is a nucleus, and this liquid is called cytoplasm. We have the DNA here. If this is strand containing adenine, thymine, cytosine, guanine, thymine, adenine, and so on, and the other strand containing, what is the complementary of adenine? Yes, thymine, and thymine containing, or the complementary of the base adenine, the complementary of cytosine guanine, the complementary of guanine, cytosine, adenine, and thymine. During the cell repairing the damaged tissue, or if you made a surgery and you want to repair your damaged tissue, or you are a young child and you supposed to grow, so DNA has to form a protein, and this protein will be translated into a trait or a character inside your body, carrying the genes or carrying the sequence of 3 nucleotides that will make the protein. So the DNA will unwind like that. The black strand far away or far apart from the blue strand. A, T, C, G, T, A. And the other strand, A, T, C. No, it's the other strand. No. T, A, G, C, A, T. So the 2 strands of DNA are separated or became far away from each other. Each strand of them are searching for nitrogenous bases inside the cell to complete the strand, making a copy for it on the RNA molecule. So we will form RNA molecule here, RNA here, and RNA here. So what is the complementary of thymine? It's supposed to be adenine. What is the complementary of adenine? Yes, thymine. But here we are forming RNA, and RNA doesn't contain thymine, it contains uracil instead. And guanine, cytosine, guanine, adenine, uracil, and thymine, adenine. On the other side, adenine, uracil, thymine, or T will form A, C, G, G, C, T, A, U. So we have here RNA strand, and on the other side RNA strand that takes a copy of the DNA, or making a copy for the DNA. After that, each strand of the DNA of the RNA, the new formed RNA, after transcription of the copy of the DNA, will be separated from the DNA and comes out inside the cytoplasm like that. Let's make a copy. A, U, C, G, U, A, and so on. And also the other strand of RNA will be separated from the DNA that took the copy before and out in the cytoplasm. In this stage, the RNA will be translated into a protein. How? As I said before, that each 3 nucleotides after each other coding for a protein or amino acid. So if we suppose that AUC coding for leucine amino acid, and the other 3 coding for valine. And after that, other 3 nucleotides coding for threonine. So here we are forming a chain of amino acids, amino acids, amino acids. So this chain will form a protein coding for a certain trait. So let's compare between the DNA and RNA to summarize what we have explained. DNA responsible for transferring the traits from the parents to offspring, and RNA transcribed or taking the copy from the DNA inside the nucleus, then transfer to the cytoplasm to be translated or to read the copies which are on the DNA or RNA to make a protein or a certain trait inside your body. So if we are carrying the traits of our parents and these traits are transferred to the offsprings, it transferred by the genes. Transferred by the genes. So DNA consists of double helix or 2 strands of DNA, and RNA consists of only 1 strand copied from DNA here and get out outside the nucleus into the cytoplasm to be translated. Also, the sugar inside the DNA is called deoxyribose sugar. Recalling the carbohydrates, deoxyribose sugar. What's the ribose sugar? Ribose sugar consists of 5 carbon atoms, and the hydrogen are the double of carbon, and the oxygen equal to the carbon atoms. This is called ribose sugar, which is found in RNA. Carbohydrates found in RNA. And the deoxyribose sugar, which is found in the DNA, consists of also 5 carbon atoms and also the same number of hydrogen atoms, but we have to decrease 1 oxygen atom to be deoxyribose sugar in the DNA. Deoxyribose sugar in the DNA. So here is another difference between DNA and RNA in the type of sugar. DNA containing deoxyribose sugar, which decreases 1 oxygen than the ribose sugar, which is found in the RNA. DNA also 2 strands and RNA 1 strand. What else? DNA nitrogenous bases are adenine, thymine, guanine, cytosine, and in the RNA the same nitrogenous bases but replace the thymine with uracil. So it contains uracil, adenine, guanine, and cytosine. Also 4 nitrogenous bases. We also said that we have hydrogen bonds between the nitrogenous bases to bind the 2 strands of DNA with each other, and we have covalent bonds that bind the components of nucleotides with each other, the sugar with the phosphate group and the sugar with the nitrogenous base. The site or the place of presence of the DNA and RNA. The DNA is found inside the nucleus, and the RNA transcribed from DNA inside the nucleus then transfers to the cytoplasm. What else? The function. DNA responsible for carrying the genetic traits, and RNA the same function plus making protein. If I ask you about the function of RNA, so it's to making the protein. Okay, I have finished the lesson, hoping you understood and try to answer the homework. See you in the next lesson, inshallah.